Hi, this is Miss Litton, and this is my wonderful Period 2 HBio class. Say hi again.
Hi.
All right, and we are continuing into the last part of chapter 23, and we are going to discuss non-Mendelian inheritance patterns. Now, I want to remind you, when you're talking about Mendel, you're talking about three laws. What are the three laws? Law of dominance, law of segregation, and law of independent assortment. Okay? Now, in everything we've done so far, if you had somebody who was big T, for a pea plant, that would be what? Tall. Okay? If you had somebody that was big T, though, they would still be tall because the law of dominance says tall is dominant over short. Okay? And you only had two letters to pick from, either big T or what? Little t. Okay? So now we're going to expand it. Instead of just having two letters to pick from, we're going to have what's called multiple alleles. So not just big T and little t, but you could have big T1, big T2, big T3, and little t. So in order to understand this, I want you to think about blood types. Tell me the blood types that you have heard about. A, B, AB, and what was the last one you said? O. Okay. Those are four different phenotypes. A, B, and O. Four different phenotypes. Before, what did we have? We had tall and we had short. Okay? So now all of a sudden we have four phenotypes. So what's going on here? So let's discuss what codes for those. If this is the phenotype, let's talk about the genotype that could create somebody who is type A blood. Here are the genes. It could be big IA, big IA. I would refer to that as being homozygous. Big I is dominant. I would say homozygous dominant. Or the other way you could be type A blood is big IA, little I. And that would be a heterozygous type A blood. Now don't look at your notes, okay? Just look up here. Could you figure out all the other genotypes from what I gave you right there? Yeah. Discuss with BioBuddy. See if your brain wraps around that. Could you figure out all the other genotypes from what I've written here? Okay, now give me another set. What do you think B is?
Big IB squared.
Big IB squared. Big I, little I.
Okay, now if you're not done yet, I'm going to show you all the possible alleles are out on the table right now. So now double check. There are no more alleles for blood than what you're seeing right there. So now figure out what the next two have to be, knowing that those are all the alleles on the table. Okay, so what's the genotype here?
Big IA,
Big IA, big IB.
That's it.
What's O? Little I, little I. Perfect. These are all the genotypes. These are the phenotypes. How many different alleles do we have here? Four. We have the big IA. What else do we have? Big IB. What else do we have? Are there any other ones? No. So how many different alleles do we have? Three. So instead of just having one dominant, one recessive, we have two variations here on the dominant. We have big IA and we have what? Big IB. Two ways that are dominant. Okay? And when you look up here, okay, we have three different alleles, but how many phenotypes do we have? Four phenotypes. Now there's another whole layer, positive and negative. I'll get to that in one more second. Okay? Now when you look up here, when you have big IA, big IB, what this means is on your red blood cells, remember how we talked about those surface markers, all the proteins, phospholipid bilayer with proteins on it. That means you have A surface markers and B surface markers if you have AB blood. If you have just A blood, that means you have only what? A surface markers. And if you have only B blood, you only have B surface markers. And if you have type O blood, you don't have either A or B. No surface markers. Now, the positive and negative has to do with another set of markers, and that's the RH, or rhesus, where it came from when they were studying rhesus monkeys, and you could be A positive or you could be A negative. If you were, hang on one second, if you're A positive, you have the A marker and you have the RH factor as well. Now, who can you give blood to? Anybody you don't piss off immunologically. If somebody has the same markers as you have, you can give them your blood. If they don't have the same markers as you, you cannot give them your blood. Okay, so if I have A blood, okay, type A blood, which I do, and I'm A negative, could I give my blood to somebody who is also A?
Yes.
Yes. They have A, I have A, I don't have the RH, so I'm not pissing them off with that. Okay? Could I give my blood to somebody who is type B blood?
No.
Okay? I can't because I will piss them off. Could I give somebody blood who has AB?
Yes.
Yes. Because they're not going to attack my A blood because they have A blood. Okay? Could I give my blood to somebody who has type O?
Yes. No.
Type O doesn't have any markers. Okay? Who would be the universal donor?
Oh.
Okay, so universal donor is going to be type O. In fact, what kind of O? O negative. O negative. They don't have A, they don't have B, and they don't have the RH. Ah. Okay, who would be the universal receiver?
AB negative or positive.
AB positive. AB positive. Nothing's going to piss them off. Okay, I had 3 hands first up originally. Yes. Yes.
I was gonna ask you, is it called O because it has like, it doesn't have any,
No, I don't think it came from that. I think it came from 0 probably.
well, it has 0 surface markers.
Yeah, I don't know. But that would be good. That'd be a good way to remember it. Yes.
So if someone were to have A positive blood and then they can't give it to A negative because
Right.
of RH.
Because of the RH. Now, let me tell you, in a pinch, if we had a zombie apocalypse, okay, or we started, another war in Iran like tomorrow, apparently, or with Mexico, apparently, or Australia, then, okay, you could actually give blood. You could give blood to somebody in a pinch one time. One time. And the reason is because it takes a while to build up your armies in response, and your blood cells are probably older already and they're going to die anyway. So you could do it one time, but then the next time they'll have memory cells ready, and if you gave them that wrong blood again, then game on. And when this has really caused problems, not now because we have the medical advice, but like somebody who's like an older grandma right now, that generation, an older grandma generation right now, I know somebody who had their first child, their next 2 children were spontaneously aborted. And the reason is because the blood of the baby was different than the blood of the mother. Now, blood doesn't cross placenta, but antibodies do. So the antibodies from her first child, whose blood was different from hers, when she gave birth, then the blood can mix a little bit when everything's going on down there, right? But then when she had her next child, also had different blood, and the antibodies crossed and clotted the blood inside the baby. And so then the baby is developed, and then the baby passed away. And her next child was the same, and then her 4th child was all right. Now, as soon as you give birth, they check your baby's blood immediately, and they already know your blood, but they'll probably recheck it. And if they are different in a way that would cause an immunological response, you get shots that are going to inhibit your immune system from having a response. So you won't develop any memory cells against it. Yes?
Is that why they used to test like you and your husband's blood type when you got
I would think so. So you would know ahead of time if there could be a problem. Yes?
So I'm a little bit confused. So the O negative you can give to any other blood type.
Yep. Because they have nothing on their surface that would trigger it. Now, if you're gonna do an organ transfer. There are other proteins on there that could make them angry, like M's and N's, and that's when they say, you know, it's not compatible, and you have to look in your family and stuff to get things like that. But as far as blood goes, you just have to worry about the A, B, and the RH factor.
Okay.
Yes?
If you have type A blood and you're giving it to AB, why doesn't the A blood like attack the B?
Because somebody who has AB blood, they won't make anti-A antibodies and they won't make anti-B antibodies because they have both. So if you're a person, okay, okay, and you have A and B antigens, they're called antigens, we haven't done the immune system yet, okay, you will not make antibodies that attach on to okay, you will not make antibodies that attach on to B or A because you would end up attacking yourself. That's why they can receive either A blood or B blood. Okay? So this is an example right here of two things for us to get from this. This is multiple alleles because we have big IA, big IB, and little I. We don't just have big I and little I. So it's an example of multiple alleles. And this right here is an example of something called codominance because both the A is expressed and the B is expressed. Codominance. They're equal. What's your question?
Yeah, regarding the positive and negative
Yes.
and the RG,
RH.
RH, yeah, can you explain that a little?
Well, I'm not holding you to that one. It's just you can think of that as one more factor. So you're either RH positive or RH negative.
Okay.
Yeah, that's just one more antigen you could have on your surface. So somebody who was, if you were, let's say, B positive, you have the B and you have the RH.
Positive, the RH positive, right?
Yeah.
Okay.
Mm-hmm. And B negative would be if you just had B and no RH.
Got it.
That would be B negative. Okay, so on your notes, multiple allelic traits defined. When a gene has many allelic forms or alternative expressions instead of just two choices. So you would have two choices if you only had big A and little a. Okay, but in this case we could have big IA, big IB, or little I. So any combination of these three alleles. So you could have any combination of these three alleles. That's all in your notes. Codominance. When both dominant allele possibilities are expressed. When both dominant allele possibilities are expressed. Okay, here are the phenotypes. Can you see them? I know it's light. Could Slate please tell me all the possible alleles you could have for these four without looking at your notes? Just call them. Did you get those? Blue, tell me what is type A blood. What are all your possible genotypes if you're type A blood? Tell me. Yes?
Yes.
Okay. And then type B blood? Yeah. AB blood?
Big I A, big I B.
And type O blood? Perfect. Okay. This then lends itself to a wonderful game called Who's Your Daddy? Okay. So if we play this, okay, let's say Jane has a baby. Okay, so Jane has a baby. Jane, that we know of, has slept with both Jim and John. Okay, that we know of. Okay. Jane's phenotype of blood is A. Don't say anything out loud, by the way. Baby's blood type is O. Jim is type B. And John is type AB. Let's find out who's going to help her buy diapers. Okay? So on your practice problems, work it out. Who is helping Jane buy diapers? Who is helping Jane buy diapers? Yeah, maybe the arrows need to be going this way because they contributed to Jane.
Whatever works.
Yeah.
Are they heterozygous?
You have to figure that out. I want everybody's genotypes. Okay, tell me, who do you think's buying diapers? Jim. I agree with you. Okay, give me Jane's genotype. Why does that have to be Jane's genotype? Yeah. How would we write baby's genotype? Little I, little I. So we know that one of these little eyes had to come from mom and one of these little eyes had to come from dad. So for sure she has to be a heterozygote in her A-ness, A category. Okay? What is John's genotype?
Big I A, big I B.
That's right. Okay, what about Jim?
Big I A, little I.
Right. Now, that is the only way. Now, this is making this assumption that she only slept with Jim. There could be a Jack in there. We don't know, right? And I'm going to tell you, blood type does not point to who the father is, but it could point to who the father isn't. Yeah, it can rule out a parent. Okay? So based on that, please riddle me this. Do your Punnett Square.
Yeah.
Hey, I got an email. I'm just telling you what it says. It says, good morning all. Just a heads up, we have the CERT team here and they are working with fire. They are located in the lunch area. Please do not be alarmed should you see or smell smoke. Heads up.
Sounds good.
This is IA. Here's a vocal word.
Did you set it up like this?
Yeah.
Indeed.
Okay. So when you do your Punnett Square, okay, this individual would have AB, here's B, here's A, and here's type O. So our phenotype ratio would be 1AB to 1A to 1B to 1O. Yeah, that's how it would work out. Good. All right. Now, that's one exception to the rule. How is it different? Because in pure Mendelian inheritance, we just have dominant or recessive. Now we have two different ways to have a dominant. We have multiple alleles, which lends itself to codominance. Now, a different thing that can happen is it looks to the eyes like blending, but it is in fact incomplete dominance. Somebody who has straight hair is H1H1. Notice variations on a dominant letter. Okay? Somebody who has curly hair is H2H2. I never forget these. Why? Because H stands for hair. Ones are straight and twos are curly. Got it? And a wavy person is H1, H2. That is incomplete dominance. Incomplete dominance. Here's another example. Red flower crossed with white flower and you get a pink flower. Black bunny crossed with a white bunny and all the bunnies are gray. Right? Now, codominance would be black bunny crossed with a white bunny and some hairs are black and some hairs are white. Red flower crossed with white flower, some petals are red, some petals are white. Okay? Incomplete dominance looks like blending. So do this for me. I have some lovely artwork to accommodate it. Say.
I've never learned to adjust.
Give me geno and phenotype ratios, no naked numbers. Geno and phenotype ratios, no naked. Did you set it up like this?
Yeah.
Okay? Now, when you get your ratios, do you notice anything about your geno and phenotype ratios? What are you noticing? They're the what? They're the same. Why is it that the genotype and the phenotype ratios are the same? Before, when we cross heterozygotes, we would have a genotype ratio of 1 to 2 to 1. We'd have a phenotype ratio of 3 to 1. Now we're crossing two heterozygotes, and the geno and phenotype ratios are exactly the same. Why is that? Blue. Tell your slated friend. Why is that? Did they say because the heterozygote has its own phenotype? Okay, now let's pretend that in pea plants, let's pretend, though it's not true, that height is incomplete dominance. What would when we cross two heterozygotes, how would that turn out then? Some would be one tall, two mediums, good, and one short, okay? But it's pure Mendelian inheritance, so it's 3 to 1 on the phenotypes. But genotype and phenotype ratios match in incomplete dominance. Okay? They match in incomplete dominance. So that's another variation. So on your notes, incomplete dominance. When a heterozygote has an intermediate phenotype between that of either homozygote. When a heterozygote has an intermediate phenotype between that of either homozygote. It doesn't look like either homozygote. Okay, answer number 2. All right, there's another set of practice problems that go along with that. A baby is going to get stolen from a hospital. Who's your daddy is going to happen. So look at those. All right. Now we're going to look at another example beyond simple inheritance, and that has to do with what I already talked to you a little bit about before when we do pedigrees. To be a female, you have to be XX. To be a male, you are what? XY. Okay. There are traits on the Y chromosome that are only on the Y and traits only on the X. So as a female, we have 2 chances at our X chromosome. Guys only have 1. Don't say anything out loud, not one word out loud. Don't ask for assistance if you need it.
That's just to see if you're
Don't say anything. Sure, I said that. I'm almost positive I did.
Oh, God, it's 13.
Okay, now, did you think it was 3?
No, I honestly saw 13.
Okay, so look at this.
What's wrong?
Red-green colorblind see the number 3. Of all the numbers, okay, you could pick. He put one with a 3 in there. Are you colorblind?
No.
Maybe. Maybe not.
It's kind of hard to see from back.
Okay. Well, let's go again. Don't help him.
Oh, wait, I can't see anything.
Interesting. We learn something new every day.
No way I get this.
Come on, Rustin. Get it right, baby. You got it. Now. All right, let's try again. Liar. Where's the hell? You got this, Rustin. Don't tell him. Don't tell him. Well, buddy, were you whispering an answer to him?
L.
Just do the best you can,
No,
sweetheart.
I got it.
You got it? Okay. Alex?
I got that one.
Okay. Now, if Rustin is having a problem, why is he having this problem? Is this his mom or his dad? His mom or his dad? His mom. What did Rustin's dad give him? A Y. What did Rustin's mom give him? An X. If that X has troubles, he only had one shot at that X, whatever his mom gave him. Does that mean Rustin's mom would be colorblind? No, she could be a what? Carrier. Could Rustin's dad be colorblind?
Can you? No, you're not. Can you?
Hypothetically, hypothetically, could Rustin's dad be colorblind? Yes, but do we care? No, because what did Rustin's dad give? Not his X, even if it might be an X that has trouble. He gave his son his Y.
Can you outline it?
Sure.
Wait, that was so beautiful. Like, look at that 5.
Why, thank you.
Whoa.
All right.
Oh, it's in them.
Good times. Okay, now, come back to me. All right, Rustin. The one thing about the colorblind allele is they write it as if it is a dominant allele. Okay, it's not. It is a recessive allele. So in this case, okay, you've got to put superscripts on the X's, just like we did superscripts on the I's. Okay, X big B, X big B. They put B like blind, but that's normal vision. Okay, X big B, normal female. X big B, little b is a carrier female. X little b, that is how a woman is colorblind. What could you tell me about any woman who has a sex-linked, and by the way, we're only talking sex-linked recessives. There are sex-linked dominant diseases, but I will not give them to you here. So don't ever worry, like, is it sex-linked dominant, okay? Only recessive diseases. So for women to have a sex-linked recessive disease, if I was colorblind, what could you tell me about my parents?
Both of them had an X that had a little
both had an X, yeah, both were
Okay.
either heterozygote colorblind or
So if I'm this, okay, what could you tell me about my dad? He has to be colorblind. Why?
He only has one X.
He gave me the X to make me a girl. So dad has to be this. Okay? What could you tell me about my mom?
She's a carrier.
She's either this, right, or this. She's either a carrier or she is colorblind to make a girl colorblind. But for a son, all you have to have for a son is a mom who is a carrier. She could be colorblind or not, but you only have to have a mom who is a carrier. Why do you, this makes sense then, why you see more men who are colorblind than women. We have 2 chances at their X. They only have 1 chance at their X. So do this for me. And so we're the same on Punnett Square. Is usually, I don't want to hear anything about it. Women are usually on top. Okay.
No, you might be right.
Oh yeah, it is normal. Sorry.
Big B's are yes. 1/8. The X, big B, big B. This is good B. This is, you're a carrier. This is good B.
So what is this like?
Did you set it up like this?
Well, I can't read it.
X big B, she's a carrier. X big B, X little b, right? And dad was normal vision, so he's X big B, Y. Can you see that number?
Yikes.
Rustin?
I think that's 45.
Yeah. Okay? So this is, I want you to hear how questions could be phrased if you ever got a club. I could say, what percentage of her children would you predict could be colorblind? 25%. What percentage of her boys? 50% of her boys.
Yeah.
Okay? What percentage could be carriers?
75%. No, it's 50%.
25%. Because to be a carrier, you have a dominant gene. It's hiding it. Okay? He's not a carrier. He has it. Carrier implies that it's hidden. Okay? So I just want to make sure you understand that. All right. So riddle me this. Flip it and you'll find carriers. And I think that's everybody in your row. Yes, please. Oh, okay, 2 more. Let's go. Justin, Navia. Oh, yeah. All right. So let's talk about this. Good job, Steven. All right. If a boy is colorblind, the boy is this, right? Oh, what is this boy? Look at me. Look at me. If a boy is colorblind, some of you picked A. His father must be colorblind. Does he need to be colorblind? No, because dad's giving the Y. We don't care what his dad is. He doesn't have to be colorblind. Could he be colorblind? Sure. Does he have to be colorblind? No. Okay? He just has to give a Y. And then on E, both B and D are correct. Okay, his mother must be colorblind or a carrier. Yes. His siblings also? No. He could have had siblings that aren't colorblind. Okay, check with your bio buddy, make sure they understand. All right. Look at this pedigree. As I told you, please listen up, you guys. As I told you, on all of your sex-linked diseases, I told you they would all be what? Dominant or recessive? Recessive. So it's going to look very much like a recessive pedigree where it skips generations. But how does this look different? What do you see in here? What's the pattern you see in here? What? It does skip. That's typical of any kind of recessive trait. But what else do you see? What? Male is more prevalent. So that's how you would differentiate between them. Okay, and you can see that here. Okay, it can skip generations, but it's more prevalent. It doesn't mean you can't have it in a girl, but if you have it in a girl, the dad has to have it too. All right, so I gave you all the notes for X-linked. Could you read through them right now with your bio buddy and make sure you understand 1, 2, 3, and A, B, and C, please? All right. Now, some examples of some sex-linked diseases. Here's Duchenne's muscular dystrophy. If a woman has, there are other forms of muscular dystrophy, but a Duchenne's muscular dystrophy is a sex-linked disease. And if a mom has Duchenne's muscular dystrophy, what can you tell me about all of her girls? They're going to at least be carriers, right? Now, the only way her girls could have Duchenne's muscular dystrophy is if she's married to somebody who also has muscular dystrophy. If there's a woman who has Duchenne's muscular dystrophy, what can you tell me about all of her boys?
They're going to have it.
They're going to have it for sure. There's no way for them to escape that. Because both of her X's are what? Bad. Okay? So these 3 boys who have Duchenne's muscular dystrophy, their mothers are either what? Carriers, or they have it. Good. Okay? And again here with hemophilia. Right? Now, the next thing on your notes is about polygenic inheritance. Polygenic. What do you hear in that? Multiple what? Polygenes, right? Multiple genes. So far what we've looked at is big T, you're tall. That's one gene, one trait. Okay? In this case, it's polygenic. Lots of different genes to give you your trait. And you see that here in most simply polygenic in your skin color. There's 2 different genes that work in additive fashion to give you your trait. Okay? To be very dark, it's big A, big A, big B, big B. Homozygous for both. To be very light, like those clear people, you're little a, little b, little b. But then you have this whole other range. So you don't have just tall or short or medium, but you have a bell-shaped curve when it comes to skin color. Okay, that's polygenic. For humans, that is true for height. We're not like pea plants, tall, short. There are multiple genes that contribute polygenic to our height. And it is not unusual for polygenic traits to be impacted by their environment. Okay? For instance, here a polygenic trait like hair color on this Himalayan rabbit, we can shave his back, which they did experiments like this. They put an ice pack on his back, let it sit there, I don't know how many different days, they kept refreshing its ice packs. And when the hair grew back in, it grew back in black. Why do you think that would be a good adaptation? When it's cold, what does black do? Yeah, absorbs more of the wavelengths of light. Okay? Now, you can get reptiles, you can determine the sex, male or female, depending on the temperature of the nest. Will determine whether they're male or female. Okay, that is not unusual, right? Is this my genotype? No, what's my genotype? Auburn with gray. Is that my phenotype? No. Did my environment influence that? Yes. Okay, so that's where your environmental, this is the whole nature versus nurture. Is it in your DNA? Okay, or does your environment influence how that turns out? Polygenic disorders like cleft lip and some emotional disorders, very much affected by their environment. And then here too are more practice problems about that. Okay, on your notes, go to polygenic inheritance. Define when one trait is governed by 2 or more sets of alleles. Two or more sets of alleles. Guys, your laughing for the last few minutes has been a little bit annoying, okay? Because it's disruptive to the people around you. 2 or more sets of alleles. And then you have examples: skin color, environmental influences, multifactorial traits. Traits controlled by polygenes are subject to environmental influences. Environmental influences. And last but not least, we've already talked about this earlier when we talked about crossing over and linked genes. We already discussed this. If linked genes are defined as all the genes on one chromosome form a linkage group. All the genes on one chromosome form a linkage group. And genes that are linked together tend to travel together to the same gamete. Genes that are linked together tend to travel together to the same gamete. 4, 4, 8. Okay, and the only thing that breaks up that linkage is crossing over. So crossing over is the only way that linkages are broken up. Crossing over is the only way that our linkages are broken up. Now, tell your bio buddy, what do you think is more likely, which linkage is more likely to get broken up? Z's and S's or Z's and G's? Discuss with bio buddy. Z's and S's or Z's and G's? Z's and G's.
How do I, Emma, where's my second?
Okay, what's more likely to get broken up? Z's and S's or Z's and G's? Z's and G's. Z's and G's. Because the crossing over can occur here, 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 and here for S's, but it can occur here, 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 and here, and here, all of these in order to break up the linked G gene. Okay? And again, there are more practice problems with this. Okay, so you want to do those as well. You might want to snap a picture of that, put it in your notes, and look at the pedigrees, and I will talk about this in the review or on Friday if you want to. Each of these, if I label these A, B, and C, which ones would work with a dominant inheritance patterns, which one would work with a recessive, and which one would work with X-linked or sex-linked? It could be all 3, it could be 2 of them. Decide. Ascribe each of these to each of these. Would this one work for dominant? Would it work for recessive? Would it work for sex-linked? Do that for all 3. Okay? And then when is the review? When is the review today? 7th period. 7th period today. Okay? And then tomorrow at lunch we are open. All right, that's it. Good job.